Column chromatography is a purification technique that separates components of a mixture depending on their polarity. In this experiment, we will be using the cis and trans isomers of azobenzene. They are loaded into a column containing a polar stationary phase and bind accordingly. A slight polar mobile phase is added which competes for binding position on the stationary phase. The mobile phase sometimes wins this competition, pushing the mixture component that is closest in polarity off the column, which we can then collect for analysis. We will be using a Pasteur pipette for the column in this experiment. It is important to notice that the one we will be using in this experiment is not the one from your drawers. It's a special pipette with thicker walls that you'll get from the stockroom. We need to create a plug that goes on the bottom of our column. The plug prevents the stationary phase from falling out of the column. Our plug is made out of glass wool. Use gloves. The plug is placed in the column and stuffed into the nose with a copper rod. Attach column to a stand. We are going to add about 2 millimeters of sand at the bottom of our column. You can use the weight paper as kind of a funnel.
Fill your column 2/3 of the way with silica gel. You want to leave about an inch's worth of space at the top. Be careful not to spill or inhale any of the silica. Now we will top the column with about 2 millimeters of sand. We set up this column the way we did because the plug and sand at the bottom prevent the silica gel from falling out of the column, and the sand at the top prevents the silica gel from splashing when you add the mixture. Prepare about 20 milliliters of a 98% hexane and 2% ethyl acetate solution. Cover your solution so it won't evaporate. Now set it aside. We'll use the solution later.
Add your azobenzene mixture to a beaker. Run a TLC to make sure that the solution has both isomers. Boil off nearly all of your methylene chloride under the hood. Remember that methylene chloride has a low boiling point and you'll need to be careful when you reach low volumes.
Remove your beaker from the hot plate just as the last bit of methylene chloride is evaporating. Once your beaker is cooled, add in 5 to 10 drops of elution solvent to redissolve your sample. The 20 milliliter elution solvent will be your mobile phase. We need to wet the column before we can run the separation. We do this by completely saturating the column with the solvent. Allow the elution buffer to run down to the bottom of the sand, then add your sample. After your sample has entered the column, continue adding elution buffer until the separation is complete.
Mix your solvent regularly. Once you feel you have collected enough trans isomer, switch collection beakers. You can see that the solution below is very light, which means we are mostly getting cis isomers out of the column. Switch your beaker again to start collecting only cis isomer. Once you begin your third collection, it is a good idea to increase the polarity of the mobile phase by making a solution with higher concentration of ethyl acetate. This will help push out the cis isomer out of the column faster. Continue running the column until all the mixture has been flushed out. Prepare and run a TLC. If you don't remember how to make a TLC plate, you can reference to the TLC video lab. Ideally, you will have one spot for your first solution. This will be the trans isomer. In your second solution, you should see 2 spots, a mixture of both isomers. And the last solution, you should see only 1 spot, which would be the cis isomer. The separation in this lab isn't perfect, so sometimes you might not see these results. When you're done, throw away your column to the glass disposal.